Hello students, welcome to today's session of chemistry. As you all know, we have started studying 5th lesson of your syllabus, that is electrochemistry. In the previous 2 or 3 sessions, we learned about the process of electrolysis. Actually, we started studying types of electrochemical cells. There are 2 types, electrolytic cells and galvanic or voltaic cells, of which we learned about electrolytic cells. The process of electrolysis takes place in electrolytic cell. What is electrolysis? It is the process in which electrical energy is used to carry out a non-spontaneous chemical reaction. And to know about it in detail, we learned about electrolysis of molten NaCl and electrolysis of aqueous NaCl. Everything about the process. That means construction of a cell, the actual reactions taking place at anode, cathode and overall result of electrolysis. And by that we came to know that this is very important process because in practice or in day-to-day life, many of the metals are extracted by the process of electrolysis. And for that, quantitative aspects are also important. Quantitative aspects, what does it mean? That means we must know how much electricity is required to produce a particular amount of a metal. To relate that, we find out one formula. That is, we learn about the derivation of that formula to calculate a mass of a product. So, that formula, derivation as well as the formula, both are important. We learn one more formula, that is the comparison between two cells if they are connected in series. That means same quantity of electricity is passed through two cells connected in series, then what will be the formula that relates both of them? We learned both the formulas. These formulas are important in both aspects, that means derivation as well as the numericals based on those formulas. As I told you, we are going to learn numericals later in one session of all the types in this lesson. Now, today we are going to learn about second type of cell, that is galvanic or voltaic cell. Because about first type of cell, that is electrolytic cell or the process of electrolysis, we just finished in the previous session. And as far as board exams are concerned, all the three, that means electrolysis of aqueous NaCl, electrolysis of molten NaCl and quantitative aspects, all of them are very important. Now, let us begin with today's session, in which we are going to learn about galvanic or voltaic cell. It is the second important aspect or second important type of cells in this lesson. Now, this type of cells are exactly opposite to that of electrolytic cells. What happens here? Here, electricity is generated through the use of spontaneous chemical reactions. So, this aspect is very important in our day-to-day life as well, because here we get electricity, that means energy, by just carrying out a simple spontaneous chemical reaction. So, this type is very important because we in our day-to-day life use these cells. That means the dry cell, you call it battery, you use for your mobiles, your everything, torch, radio. Those batteries or dry cells are kind of galvanic or voltaic cell. So many of the batteries or the galvanic cells are important in our day-to-day life. So what actually happens there, how the electricity is produced, that we are going to learn in this type of cell. So let us start. A galvanic cell is made up of two half cells. These are very important basic things. So we require two different half cells. One cell where oxidation takes place, another where reduction takes place. That means two different half cells are first prepared and they are then connected. So a galvanic cell is made up of two half cells. Each half cell consists of a metal strip immersed in the solution of its own ions of known concentration. Understood? Now, concentrate on one half cell so that we can prepare two different half cells. How it is prepared? Just take any metal strip, that means copper, zinc, aluminum, silver, whatever, and that metal strip is immersed, that means dipped in the solution of what? In the solution of its own ions of known concentration. Let us take an example. Suppose we have taken a strip of zinc metal. Then that strip we should dip in the solution of 1 molar of aqueous solution of zinc ions to form an half cell. Now where we will get zinc ions? Ions, how they are produced? By the process of dissociation. And where that process takes place? Zinc ions you cannot get separately as zinc ions. They must be produced in the solution itself. How we prepared Na plus and Cl minus ions? We dissolved that salt in water. In the same way, we have to take any salt, and if we dissolve it in water or in an aqueous medium, we get different ions. One is positive, another is negative, because salt is formed from cation and anion. That means if we want to prepare a half cell by immersing zinc strip, that means the solution in which we are going to dip that zinc strip must have zinc ions. And how zinc ions are produced? By dissolving the salt of zinc. That means zinc sulfate, zinc chloride, like that. If we dissolve 1 mole of zinc sulfate salt in water, you will get 1 mole of zinc plus 2 ions, that is Zn plus 2 ions. And if that means we know the concentration of zinc ions, and we are going to dip a strip of zinc metal in that solution, and that becomes your half cell. That means, understood, you have to take any metal strip. Suppose we have taken magnesium, then dip in the solution of magnesium sulfate. Take a strip of silver, Ag, and dip it in the solution of any concentration of known concentration, we say 1 molar suppose. Then dip silver strip in silver nitrate, dip magnesium in magnesium sulfate, like that, aluminum in aluminum nitrate. So we have to take any salt, dissolve it in water or in an aqueous medium, and dip the strip of that metal in that solution, and that becomes your half cell. Understood? Now, we require two different half cells to construct a galvanic cell. Now, next point. It follows that two metal plates and the solutions of their ions with known concentrations are required for the construction of a galvanic cell. Two half cells are constructed by immersing the two metal plates in solution of their respective ions placed in separate containers. In electrolysis, we have taken only one container because we are going to carry out the electrolysis of one salt only. But here, in galvanic cell, we are going to carry out a spontaneous chemical reaction. For that, we require two different containers in which two half cells are constructed. How the half cells are constructed? By dipping the metal strip in the solution of two solutions of their own ions. Now, these two separate containers or half cells so constructed are combined together to form the galvanic cell. Now these two half cells should be connected, then only the reaction will take place. So they are combined. They are combined in two ways. That means to externally as well as internally to complete the circuit. How they are combined externally? These two metal plates. Now we are going to combine metal plates or strips and metal solutions, both. The two metal plates are called electrodes and they are connected through voltmeter by a conducting wire for transfer of electrons between them. In electrolysis, how we connected battery to two metal electrodes by using wires, in the same way we are going to connect these two metal electrodes or metal strips by conducting wires through voltmeter. Why voltmeter? Voltmeter will show you deflection, and when a reaction starts and if voltmeter shows deflection, that means current flows, that means chemical reaction has produced electricity by transfer of ions. And to know that, we have to connect these metal electrodes by voltmeter. Now we have to connect two solutions. These to complete the circuit. To complete the circuit, the two solutions are connected by conducting medium through which cations and anions move from one compartment to the other, and this requirement is fulfilled by a salt bridge. So a new word, salt bridge. That means two metal electrodes, these two containers are connected by using salt bridge. So salt bridge we are going to learn about. Now that means the figure will look like this. What we did? We have taken this one half cell, this is another half cell. How the half cell is constructed? We have dipped the metal strip in the solution of its own ions. That means we have taken copper strip and dipped in the solution of copper sulfate. In the same way, we can take zinc strip dipped in the solution of zinc sulfate. Two separate half cells are connected externally through voltmeter by conducting wire. That means two wires are connected and in which voltmeter is in between, it is attached. Now, these two half cells or the solutions are connected by using this salt bridge. As it connects, it is called bridge, but it actually has salt. Why this salt bridge is taken? That means through this salt bridge we can say the cations and anions can pass from one container to another, avoiding mixing of two solutions. Hence, this will make a complete galvanic cell which is connected externally as well as internally so that the circuit is complete. We are going to see actual process. But before that, a basic thing you should understand that how the cell is constructed. We can take any salt solution here. Only the thing is we have to dip the strip of that ion only. Hence, we can construct a cell. Other things will remain same. That means this connection, internal and external will remain same. Only the containers will be different. And variety of type of galvanic or voltaic cell we can prepare. Now, let us study about salt bridge. What is salt bridge? Salt bridge is required to connect two solutions. So in galvanic cells, two solutions are connected by a salt bridge. It is an U-tube. It is actually a U-tube containing a saturated solution of an inert electrolyte such as KCl, or there is potassium chloride, or NH4NO3. That means ammonium nitrate. And these salts are in 5% agar solution. That means you have to take any salt, generally strong electrolyte is used. This salt is taken in the solution form and the solution is made up of 5% agar. Agar is a kind of gel or it is a powder. When it is dissolved in water, it gets converted into jelly material. And this salt solution is taken in that agar medium. Now, The salt bridge, how it is prepared then? It is prepared by filling U-tube with hot saturated solution of the salt. That means KCl, suppose we have taken KCl, then it's hot and saturated, that is concentrated. Solution is taken and agar-agar solution is also taken, and all this is mixed, that is hot saturated solution of the salt and agar-agar, 5% agar-agar, these two are mixed. It is taken in hot condition because the cooled solution sets into a gel which does not come out in inverting the tube. We are taking, actually I am saying that it is U-tube, but for connecting two cells it is inverted, that is inverted U. Hence, when we invert it, the solution may come out. To avoid that, agar is taken. When agar is cooled, it gets converted into jelly material so that it will not come out. And the mouth of the salt is closed by using glass wool. Now, the salt bridge is kept dipped in distilled water when not in use. That means the salt bridge is actually like this. The tube is hollow, containing saturated KCl solution plus agar gel. And when it becomes cool, that agar will convert into gel. And the mouth is closed by using glass wool plugs. The plugs, that means like cotton plugs we take, like that fine strips are glass or removed, and the mouth is closed by using glass wool plug. So this is, when not in use, it is dipped in distilled water, and otherwise when in use it is dipped in the two different half cells and the half cells are connected. So this is salt bridge. What actually salt bridge does, that means functions of salt bridge we will see. The salt bridge serves as number one, it provides an electrical contact between two solutions. The basic function is to connect them. Electrical contact between two solutions, and thereby completes the electrical circuit. Secondly, it is useful in preventing mixing of two solutions because we have taken two different salt solutions. So their mixing must be avoided. The third one, it maintains electrical neutrality in both the solutions by transfer of ions. It is very helpful. How? We will see when actually we learn about any cell. But it maintains electrical neutrality. That means by transfer of ions through it from one container to another, we can maintain electrical neutrality. So this is all about functions of salt bridge. So understood about the cell? We have constructed a galvanic cell by two half cells. These two half cells, they contain metal strip dipped in the solution of its own ions of known concentration. These two metal strips connected externally by using electrical wire in which voltmeter is attached to know whether actual electricity flows or not. And these two half cells connected internally so that the ions pass through that and the circuit will be complete by using U-tube, and that is called as salt bridge. So actual galvanic cell we are going to learn in the next session. So time for assignment, and this is the assignment number 10. Write a note on a salt bridge. That means you have to write the basic construction of salt bridge, its figure and the functions. So a question for 2 marks has been asked in board exams about the functions. Sometimes write a note on salt bridge. So write down the note on salt bridge. Thank you.